Hello everyone. So today we are going to continue the chapter number 5. Hello everyone. Today we are going to continue the chapter number 5, that is molecular basis of inheritance. This is theory part 9. In this section we are going to cover the NCERT section 5.7, 5.8 and 5.8.1. So in today's class what we are going to see actually is that today we are going to talk more about translation process, like how this translation take place, then some gene expression system, and yeah. So today we are going to talk about that only. Disclaimer. So what are going to be today's learning objectives? So first we will understand protein synthesis steps. So what are the protein synthesis steps? What are the steps that has been taking place in translation, that is initiation, elongation or termination. Then we will identify some key components that are used in that process, in the process of translation, that is ribosome, tRNA and mRNA. We have talked about tRNA, mRNA, their structure, and even we talked about ribosome. But today what we're gonna focus is that how they are used, in translation. Then we will explain you some gene expression regulation mechanisms. Then we will describe transcriptional, translational, and post-translational control. Then we will understand the lac operon structure and function. Later, we will analyze the positive and negative regulation in the operons. So, what is going to be today's index? So, index will look like this. First, we will cover the translation part, which is NCERT 5.7 section. Then we're going to talk about regulation of gene expression, which is 5.8 section of NCERT. And in today's class, we're going to talk about its subsection, that is the lac operon. Then I'll give you the summary of all the slides or all the things we have studied, the main points that we have studied in today's class. Then there will be some NCERT based question that we're going to solve together, some keynotes, followed by interesting questions that you guys gonna solve yourself. So what is translation? Okay, what is the definition of the translation? We have listened this word again and again and again, and today we are going to understand its to its depth, like what actually happened in translation and all those things. But let's talk about the definition first. So if we want to talk about definition, so the definition of translation is that the process of polymerization of amino acids to form what? To form a polypeptide. So the process from which this mRNA turns into protein. How? The process, with the help of this process of polymerization of amino acid to form this polypeptide, is what we call translation. The order and sequence of this amino acid are defined by the sequence of bases in the mRNA. So, what is going to be the order and sequence of this amino acid? That who defines or who know? That is defined by the sequence of bases that are present in what? That are present in mRNA. That's what we even studied in the yesterday's class, that if there is any alteration in the nucleotide sequence of DNA, how it is going to affect the polypeptide, because everything is interconnected. Now, the amino acid are joined by a bond which is which is known as peptide bond. This from here you guys can answer NCERT exercise 14C. So they are telling that amino acid, we know that what is polypeptide. Okay, what is protein? So it is a chain of amino acid. Then definitely these amino acid had to be linked together, has to be joined together. So they join by a bond which we called peptide bond. So This process of polymerization of amino acid to form a polypeptide, this figure is showing what? It's showing translation. We are going to understand each and every component which is like, taking part in this whole translation thing, but that you guys will understand after few slides. Now first we will talk about like peptide bond, peptide and polypeptide. So what is actually peptide bond? I told you, like, when two amino acid are joined, they are joined by peptide bond. But what actually is peptide bond? It is a chemical bond between two amino acid. How this bond forms? So this bond forms between carboxylic group of one amino acid and amine by joining with the amine group of another amino acid. If you talk about peptide, what is a peptide? Peptide is a short chain of amino acid, typically from 2 to 50 amino acid. Amino acid are linked by what? Amino acids are linked by peptide bonds. So here you guys can see. So as I told you how this peptide bond forms. So carboxylic group of one amino acid at the C terminus forms bond with amine group of another amino acid at N terminus. Okay, at their N terminus, and this is what we call peptide bond. So what will happen? One water will released, as you guys can see, this H2O, 2H and 1O got released, and now what like formed here? A peptide bond formed here. So this is how a peptide bond form between two amino acid. Amine group of carboxylic group of one amino acid joined to whom? Joined to amine group of another amino acid, and this is how a peptide bond is formed. So now we will talk about the machinery which is required for what? Which is required for translation. First is mRNA, that is messenger RNA. From here you guys can answer NCERT exercise 8B. So what is an mRNA? So if we talk about the translation things, the mRNA is the template that is carrying what? That is carrying the genetic code. If you guys gonna talk about mRNA structure, it contains what? It contains translational unit. The translational unit, what is translation unit? The unit which is going to be got translated, which is going to be become a protein, which is going to translate to this amino acid, and this amino acid formed by the peptide bonds going to make what? Peptide, a protein. So this mRNA structure contains what? A translational unit which is flanked by what? Which is flanked by a start codon, and we got to know in yesterday class what was a start codon. AUG is a start codon. It acts as a start codon as well as it codes for methionine amino acid and stop codon. So what they are saying, this mRNA structure look like what? I'll let you, like, in this slide only we have a picture. I will explain you there more. Then mRNA also has UTRs. Now what are UTRs? UTRs are the untranslated regions, the regions that are not going to translate. Now the UTRs are present at both 5'N, which is before the start codon, as well as at 3'N, which is after the stop codon. These UTRs are important for what? These UTRs, these untranslated regions, are important for translation efficiency. Means this region is not going to translate, but this region is important for the translation efficiency. So now let's see the whole this thing. Here you guys can understand it more properly. Like, see from here. DNA is transcribed into mRNA. This is something we know. This was a DNA. It got transcribed into what? It got transcribed into a mRNA. Now what mRNA has? mRNA has the genetic code. Now this mRNA is translated into what? Into amino acid. Each codon, this triplet correspond to what? Correspond to one amino acid. Now these codon will code for what? These codon will code for amino acid. That amino acid form what? Polypeptide chain. This polypeptide chain fold into protein. This is how this whole process taking place. Okay, now if we talk about the structure of mature RNA, what was the structure of mature RNA? mRNA. So as we see, we have known, like we have studied in the transcription that the 5' end has what? This methyl guanine triphosphate. 3' of this mRNA has what? Polyadenylated tail. Okay. Now, what this mRNA part have now? We know it has start site, start codon. It has this stop codon. It has the coding region, means it has the regions which was having what genetic code, and it has some untranslated region, that region which is not going to translate it. So this is the whole structure of what? This is the whole process of this. This is kind of whole central dogma, that how DNA convert to mRNA and mRNA into translate into protein, and this is the structure of a mature mRNA. Okay, so this is how it gonna work. Now, so first for this translation thing, we have studied about mRNA. Now it's time to understand ribosome. Okay, so if we talk about ribosome is what? It is the workbench for protein synthesis. So this is the place where this protein synthesis is happening. Okay. This is the cellular factory responsible for what? For protein synthesis. Ribosome consists of structural RNAs and about 80 different proteins. So ribosome consists of what? Structural RNAs and about 80 different proteins. It exists as two subunits. So ribosome, irrespective it is like prokaryotic ribosome or eukaryotic ribosome, it is like what? It is having two subunits. One large subunit and one small subunit. The small okay, in its inactive state. If we talk about the catalytic activity of ribosome, from here you guys can answer NCERT exercise question number 9. If we talk about the catalytic activity, the ribosome, specifically which ribosome? The 23S rRNA in bacteria act as what? Act as catalyst. And what we call it? We call it ribozyme. It catalyzes what? It catalyzes the peptide bond formation. So here you guys can see, what is this? This is mRNA, this is a ribosome. We will study in upcoming slides that you know how this ribosome help in translation and all those things. This ribosome has what? This ribosome has one large subunit and one small subunit. If we talk about the catalytic activity of the ribosome, so 23S rRNA, okay, in bacteria, it acts as catalyst, which we call as ribozyme, and it catalyzes what? It catalyzes the peptide bond formation. This thing that this is mRNA, this is small subunit, this is tRNA docking site, that you guys will understand in after 2-3 slides. Now, we talked about what? We talked about in the machinery required for translation. We talked about mRNA. We talked about ribosome. Now it's time to talk about tRNA, transfer RNA. We have studied about its structure and its function in last class, but now we will understand its role in translation as well. From this part, you're going to answer NCERT exercise 8 part B. So what is tRNA? It is the adapter molecule for amino acid. We have studied it in detail in yesterday class. It carries what? It carries amino acid to ribosome. Attachment of amino acid, we have studied about that acceptor end. That attachment of the amino acid with tRNA referred to as what? Charging of tRNA. Means when the amino acid will go and will bind to this acceptor end of tRNA, what we can say, we will not say that this is attachment of amino acid. We will say that this is charging of tRNA. tRNA is now charged. Why? Because it has the attachment of amino acid. So this is what we were talking about yesterday. We have seen what? We have seen this anticodon loop, which has what? Which has nucleotide, which has bases that are complement to this codon part, which is on mRNA. And we talked about this acceptor stem, which is going to have where this amino acid will go and attach. And this is what we will say, this is whole structure is known as, because it has amino acid, amino acyl tRNA, and we can say that this tRNA is charged. Now next, So we have talked about the different machinery that we will require in the translation process. It is mRNA, ribosome, and tRNA. Now we will understand the steps that are involved in translation, the process flow in translation, and this from here you guys can answer NCERT exercise question number 14C. So let's start. First, charging of tRNA, what we also called amino acylation, then initiation, elongation, and termination. These are the steps involved in translation. We're going to talk about each. First is charging of tRNA, that is amino acylation. So what they are saying, there is saying that formation of peptide bonds requires energy. Thus, amino acid are activated in the first phase. Because we know after the amino acid will be formed, what we need to do? We need to join those two amino acid with the help of peptide bond. We have studied in the second slide that what is peptide bond. Peptide bond is when two amino acid means carboxyl group of one amino acid and amine group of another amino acid join together, okay, by the release of water. This is what we make peptide bond. So the formation of those peptide bond between the amino acid, it requires energy. Thus amino acids are activated, therefore acids are activated in the first phase. Activation of amino acid, how it happens? Amino acids are linked to their cognate what? tRNA. This activation process is carried out in the presence of ATP. Activation process also called charging of tRNA or amino acylation of tRNA. So now in yesterday class, what we have seen, what the use of the tRNA, that it has this anticodon, which go and bind to this complementary codon part. Then the amino acid with respect to that codon comes and attached to this acceptor end. So why it is important? Why the charging of this tRNA amino acylation is important? Because we know that the formation of peptide bond require energy. Therefore, this amino acid are needed to be activated. How this activation of amino acids occur? So this amino acids are linked to what? To their cognate tRNA. Okay. The tRNA for which they have this anticodon. This activation process is carried out in the presence of what? ATP. ATP is required in this process. This activation process also called charging of tRNA or amino acylation of tRNA. Now, this amino acylation of tRNA happens with the help of the enzyme, what we call amino acyl tRNA synthetase. Okay, what will happen? This is the tRNA transfer RNA. This enzyme help this amino acid go and bind to this tRNA. And what is this enzyme is? Amino acyl tRNA synthetase. You guys can see what happened. This tRNA go and bind to enzyme, then this came, and this charged tRNA has formed, which we also call amino acylation of tRNA. Okay. So this was about the charging of the tRNA. So what happened in this tRNA charging? So first we know what is the structure of this tRNA. tRNA has what we need to understand about tRNA, that tRNA has one anticodon loop. In this, it has bases which is complementary to what? Which is complementary to the codon which is present on mRNA. And with respect to that anticodon and codon only, the amino acid will come and bind to its one end, okay, acceptor end, as it will come and bind there. So how this process is taking place, and this process is what it is called? It is called the amino acylation or charging of tRNA. So what will happen? This amino acyl tRNA synthetase, which is an enzyme, this will help in doing what? Helping attaching this amino acid to its respective, to its cognate tRNA. And this structure and this whole is called amino acylation of tRNA or charging of the tRNA. Next step is initiation. Now what happened? When the small subunit encounters an mRNA, the process of translation of mRNA to protein begins. So now what will happen? We know that ribosome has two subunit. Okay. So first, the small subunit encounters mRNA. Okay, the mRNA on which there is genetic code, and the process of translation of mRNA to protein thus begins. The small ribosomal subunit bind to the mRNA at what position? At start codon. We know that this is the point from where translation will start. So the small ribosomal subunit will go and bind to mRNA at the start codon. Now what will happen? The initiator tRNA, the tRNA which is carrying methionine. Why? Because it has the anticodon for what? For this AUG. Okay, it will be have U A C, the anticodon for the start codon, that initiator tRNA with methionine at its acceptor end and anticodon end, it has codon which is complementary to the start codon, will come, recognize the start codon and binds to it. Okay. Now, the large ribosomal subunit then binds to this complex, forming what? Forming the complete initiation complex. So now you guys will understand this more from this diagram. So what happened? So this was our mRNA. Okay. Now what happened? First, the small subunit came. Okay, encounter this mRNA and binds to the mRNA at which position? At the start codon. Okay, at this AUG codon. Now what happened? The initiated tRNA, this which has methionine and anticodon for this AUG start codon, it will come, recognize the start codon and binds to it. After this will binds to, this binds to this mRNA, this AUG. What happened? The large ribosomal subunit will come, bind, as you guys can see, the large ribosomal subunit came and bind, and now this complete complex is called initiation complex. Okay. After this initiation, then there will be the process of elongation. Complexes of amino acid linked to tRNA molecules bind to the mRNA. We know that every amino acid has its specific tRNA, and now tRNA will come and bind to the mRNA, and this formation of amino acids will start. Binding occurs how? Binding occurs by a complementary base pairing between the tRNA anticodon and appropriate mRNA codon. Now ribosome move along the mRNA codon by codon. Amino acids are sequentially added to growing polypeptide chain. DNA information, which was represented by mRNA, is now translated into a specific polypeptide sequence, what we call protein. Now we guys will understand this structure. What happened? As I told, in initiation, what happened? First, small subunit of ribosome came. Okay. Then what happened? After this small subunit of ribosome came and bind to this start codon of mRNA. The tRNA, which is specific for methionine and it has anticodon for this start codon, it came and it bind to that part. Then the large ribosomal complex came, large subunit of ribosome came, and it whole now make a what? Initiation complex. Now what will happen? Now elongation step will happen. Now in this elongation step, what will happen? The tRNA, okay, the tRNA molecules which are linked to their amino acid will now start coming. Binding occurs between the complementary base pairing between whom? Between the tRNA anticodon and the appropriate mRNA codon. And this ribosome, the whole ribosome structure, will move along the mRNA codon by codon. Just like moving from one home to another to another. Just like in sequence, this ribosome is moving from mRNA codon by codon. And sequentially, what is happening? The whole growing polypeptide chain is growing. Okay, so this is the whole process. You guys can see this is what? This is an mRNA. This is the ribosome. This is the tRNA with its specific what? With its specific amino acid. Okay, and it is growing like this. So this is how the translation process occur. Then what happened? Now after this elongation step, Now the last step will be termination. How the termination will happen? When the ribosome reaches a stop codon, that is UAA, UAG, UGC on mRNA, and we know that there is no tRNA for stop codon. Okay, so when the ribosome reaches a stop codon on the mRNA, no tRNA can bind. A release factor bind to the stop codon. This triggers the hydrolysis of the bond between polypeptide and tRNA. This hydrolysis allows to release complete polypeptide. After that, ribosomal subunit dissociate from the mRNA. So technically, what happened in this last step after the elongation? What happens? This ribosome reach a stop codon, and now translation has to stop because there is no tRNA. No tRNA can bind to now mRNA. So now what will happen? You guys can see here in the figure, a release factor, a protein bind to what? Bind to stop codon. This trigger what? This trigger the hydrolysis of the bond between the polypeptide and the tRNA. Now because of this hydrolysis of the bond between the tRNA and polypeptide, what happened? Polypeptide will release, complete polypeptide will release, as well as this ribosomal subunits also dissociate from the mRNA, and this is how the translation process will complete. So now we came to the next unit, that is the next section, 5.8, regulation of gene expression. So gene regulation is the process of controlling when and how our genes are expressed. So what is a gene regulation? It is a process which will decide, which will control when and how our genes are expressed. So at what time the genes are going to be expressed, at what is the proper time, every condition when this gene has to express, this all thing is considered under gene regulation. So in eukaryotes, gene regulation can occur at multiple levels. So when we're talking about eukaryotes gene regulation is occurring at multiple levels. So levels of regulation in eukaryote. So what are the levels of regulation that occurs in eukaryote? First is transcriptional level, controlling the formation of what? Controlling the formation of primary transcript. Then processing level, this is the regulating like splicing and all those process. Then transport level, regulating the transport of mRNA from nucleus to the cytoplasm. Translational level, controlling the synthesis of protein for mRNA. So how we can regulate eukaryote? How we can regulate this gene expression of eukaryote? We can regulate it at several levels, multiple levels. First level, for example, we can regulate it at transcriptional level. How? By controlling the formation of primary transcript. Then processing level, how it can be regulated? It can be regulating by regulating some process like splicing and capping and all those things. At what level of transport level it can be regulated the transport of mRNA from nucleus to cytoplasm. And if we talk about last level, that is translational level, so this can be controlled by the synthesis of protein from mRNA. So at these levels, gene can be regulated at the multiple levels in the eukaryote chain. Now, transcriptional initiation rate is the main control point. So when we would now, if we will talk about prokaryotes, this we talked about eukaryotes. In eukaryotes, we can regulate, not we can, means gene expression can be regulated at various level, okay, multiple level that we have studied. But if you guys talk about gene expression in prokaryotes, it can be regulated, the main control point is transcriptional initiation rate only. Okay. RNA, and who governs this? Who governs this transcriptional initiation rate? It got governed by RNA polymerase activity at promoter. We have studied about this transcription, that how RNA polymerase will go and bind to promoter site and all those things. So this activity is going to govern this transcriptional initiation rate. Now, accessory proteins, other proteins like activators or repressors, they can regulate RNA polymerase activity by affecting its ability to recognize start sites. So, we are telling in prokaryotes, transcriptional initiation rate is the main control point. Okay, now this transcriptional initiation rate is governed by whom? Is it governed by the RNA polymerase activity at the promoter site? But here now we are saying there are some accessory proteins like activators or repressors that can regulate this RNA polymerase activity. How? By affecting its ability to recognize sites. So now we will talk about something. First is operators. It is the DNA sequences near promoters. Protein interaction with operators control RNA polymerase accessibility. Because we talked about there are accessory proteins that can regulate the RNA polymerase activity. So now we are talking about some proteins, some accessory proteins that can do so. So for example, operators. So this is not a protein. Actually, this is what? This is a DNA sequence. It is the DNA sequence near promoter. Protein interaction with operators can control RNA polymerase accessibility. So here what it is saying that we talked about what? We talked about that there are accessory proteins that can regulate RNA polymerase activity. How? By affecting its ability to recognize the start size. Now here we are talking about operator, which is a DNA sequence near the promoter, and this operator can interact with protein, okay, and that can control the RNA polymerase accessibility. That how this RNA polymerase is accessible. Operators usually bind repressor protein. So what this operator do? This operator usually bind repressor protein. Each operon has specific operator and repressor. For example, lac operator and lac repressor. So each operon. Now you guys can ask, what is operon? Okay, because this word now we are gonna use a lot of time, that operon or lac operon. So you guys first need to understand what is this operon mean. So operon is nothing, it's just a unit, okay, which is made up of linked genes, which is thought to regulate other gene, okay, which is responsible for protein synthesis and all. So this is nothing, this is a unit made up of linked genes, which is thought to what? Which is thought to regulate other genes which are responsible for protein synthesis. So now here we are saying that each operon has specific operator. So means whole this unit has specific operator and repressor. Because we are here saying what? We are saying that operators are the DNA sequence which are near what? Which are near promoter, and if these operators interact with proteins, it can control the accessibility of RNA polymerase. And operator what actually do? It usually bind to repressor protein. I know it sound quite confusing here, like what is operator, what is repressor, what is any like what is operon, what is promoter. Every word is like come together, but believe me, from next slide when you will understand, when we talk about lac operon and all those things, you guys can understand it much clearer. So here what you guys need to understand, main focus on that. If we talk about gene expression in prokaryote, the main control point is transcriptional initiation rate. Okay, this rate is governed by what? This rate is governed by RNA polymerase activity, because we know that promoter will go bind to RNA polymerase. RNA polymerase will go and bind to promoter, and this whole transcription will start and all those things. So governs by what? RNA polymerase, but there are some accessory proteins like activators or repressors that can regulate this RNA polymerase activity. Okay, by affecting what? By affecting its ability to recognize the start sites. Now, what operators are? Operators are nothing, they are the DNA sequences which are near promoter. Protein interact with this operator and can control RNA polymerase accessibility. Operator do what? Operator usually bind repressor protein. Okay, and each operon has its specific operator and repressor. This is a line which you guys will understand later. But I told what is a operon. It is a Unit where there is linked genes, that can control the other genes which are coding for protein synthesis. So now let's talk about the lac operon. This is NCERT section 5.8.1. So what is this operon concept? So as I told that operon is a unit of prokaryotic gene expression. It includes a cluster of genes that are under the control of a single of promoter and regulatory genes. So what is an operon? It is a unit of prokaryotic gene expression, and it includes a cluster of genes that are under control of what? That are under the control of a single promoter and regulatory genes. If we talk about what are the elements of operon, so it will be promoter, it will be operator, and then it will be structural gene. The operon system is very common in bacteria. Examples are lac operon, TRP operon, ERA operon, his operon, val operon, etc. This is an operon system, but you guys will understand it more when we talk about lac operon. So now what is lac operon? First of all, let's talk about who discovered lac operon. So Francois Jacob and a biochemist Jacques Monod were the first to elucidate a transcriptionally regulated system. They describe a model for gene regulation in what? In Escherichia coli bacteria related to the lactose metabolism, and that's why it was called lac operon. So these are the scientists. These scientists has discovered this, transcriptionally regulated system, and this is in the E. coli bacteria, and it is related to lactose metabolism. So now if you talk about the structure of lac operon, what is the structure of lac operon? In lac operon, a polycistronic structural gene is regulated by a common promoter and regulatory genes. So we know that, from first or second class we have learned about this polycistronic or monocistronic gene. So polycistronic, that is going to code for multiple proteins, and mono was that is going to code for one protein only. And we got to know from there only that prokaryote have mostly polycistronic structural gene. So what happens in a lac operon? In a lac operon, this polycistronic structural gene, which is going to code for multiple proteins, is regulated by a common promoter and regulatory genes. And how? That we are going to know. It consists of what? It consists of a regulatory gene, promoter, operator, and 3 structural genes. So here is the structure of the lac operon. What is in this? A regulatory gene, a promoter, a operator, and the 3 structural gene. And this is whole what we call lac operon. This from promoter from this is called lac operon. Now we will talk about the structure of lac operon. First is regulatory gene, which we say I gene, codes for the repressor protein of the lac operon. I does not here refers to inducer, rather it is derived from the word inhibitor. The repressor protein is continuously produced. This production is not regulated by the operon. We say it is constitutively expressed. So first we talk about what? First we talk about the regulatory gene. So what is here a regulatory gene? I gene. This code for what? This code for repressor protein. Okay, repressor protein of whom? Repressor protein of this lac operon. And here I doesn't refers to inducer, rather it is derived from which word? Inhibitor. This repressor protein is continuously being produced. This production is not regulated by this operon. Okay, and that's why you guys can see this, that this is not even, part of lac operon. This is the regulatory gene, and this is not regulated by the operon. We can say it is constitutively expressed, means it is continuously being transcribed. It's been expressed. Then we will talk about promoter. This is the binding site for what? For RNA polymerase. We have studied about this promoter, this sigma factor binds to this RNA polymerase. Then RNA polymerase will go and bind to the promoter, and then how will transcription start? Then is operator. This is the binding site for what? It is the binding site for the repressor protein that regulatory gene is transcribing and encoding. It is located adjacent to the structural gene. So you guys can see what is this. This is the promoter. This is where this is the binding site of the RNA polymerase. Then there is this operator which we have studied. What it is it? It is DNA sequences. Okay. And here where this is the place where the repressor protein will come and bind. Okay. And it is located adjacent to what? Adjacent to the structural genes, the genes which are going to encode for the proteins. Then comes the structural genes. These are the three structural genes. All three gene product in lac operon are required for what? Metabolism of lactose. So these three genes, these three structural genes are going to code for the proteins which is required for what? For the metabolism of lactose. Here is the picture. And we have talked about the first what we have talked about. We have talked about the regulatory gene that has been shown by lac. Then there has been what? There has been the promoter gene, then there has been the operator gene, and then there has been what? This three structural gene. Okay, here you guys can see in the figure also, this is what lac, the regulatory gene, the gene which is going to produce what? This is going to produce the repressor protein. Okay, then this is the promoter part. Okay, then this is what? This is the operator which is just adjacent to the promoter, and then this is what? This is the structural gene. Now, if we talk about the function of the structural genes, let's see one by one. First is Z gene. Okay. First is Z gene, which and it's going to produce which product? It is going to produce beta galactosidase, beta gal. It is primarily responsible for hydrolysis of what? Hydrolysis of disaccharide. Then, thus it hydrolyzes the lactose into its monomeric unit, that is galactase and glucose. Then comes Y gene, which is going to produce permease. It increases what? It increases the cell permeability to the lactose. Then A gene is going to produce transacetylase. And what its function is, it transfer an acetyl group from acetyl coenzyme A to galactosidase. So these are the three products that has been formed by the structural gene, and its main function is in like lactose metabolism. Now, how this regulation of lac operon occurs? So here lactose acts as inducer. Lactose is the substrate for the enzyme beta galactosidase. It regulates switching on and off of the operon. Thus, lac operon is also called inducible operon in E. coli. So here what you guys need to understand is that lactose act as inducer in this lac operon. Okay, lactose is what? Lactose is the substrate for the enzyme beta galactosidase for which that gene Z has been encoding. Okay, it regulates the switching on and off of the operon. This lactose will decide like when this operon will switch on and when it will be switched off. And that's why this lac operon is also called inducible operon in E. coli. So what is the function of beta galactosidase actually? So it converts this lactose into glucose and galactose. Okay, hydrolysis of lactose it does. That we have also studied in that table. Then if we talk about the Y gene, what it do? Leaky expression of lac operon, a low level of lac operon expression is always needed. Without it, galactose cannot enter the cell. This is because the lac operon produces permeases which is required for lactose import. So what we are not understanding, what you can understand from this slide is that there's always been a leaky expression of lac operon. A low level of lac operon expression is always needed. Without it, lactose cannot enter the cell, and this is the only reason why lack operon produces permeases. And you know that we have studied that permeases will do what? It will make cell more permeable to for lactose import. Then come the case. Now it's very important to understand from here how the regulation of lac operon happened. First, the inducer. We told now that inducer is the lactose is the substrate. It is an inducer. What will happen if this inducer is present? Lactose inducer is transported into cell through the action of permeases. We know we need permeases. Why? To make it to import the lactose inside the cell. Lactose then bind to the repressor protein. This binding causes a conformational change in the repressor, inactivating it. The inactivated repressor cannot bind to the operator. So, what is happening here? Understand it properly and understand with all your focus. First, this is the case when the inducer, when this lactose is present. Okay. So now what is happening? This lactose, this inducer is transported into the cell, which from which action? With the help of action of permease, that this gene Y, the structural gene Y, is been encoding. Okay. Now what happened? Now this lactose, okay. Now this lactose, here is the lactose, you guys can see. Right now we are understanding this position here. Pay attention at here. In the presence of inducer, this is the case study 1. So now we have this inducer, we have this lactose into the cell. Now what will happen? This inducer, this lactose, will bind to this repressor protein. This repressor protein. What is this repressor protein? This is the protein which has been, actively producing by which? This regulatory gene, which we have talked about, LAC-I, this I gene. Okay. This is producing this repressor mRNA, and this mRNA is translating into what? Into this repressor protein. Now what happened? Inducer, lactose is present, and this inducer will come and bind to what? Bind to this repressor protein. Now because this repressor protein now get binded with inducer, now this repressor will, this binding will cause a conformational change in the repressor, and now this repressor become inactivated. Now what will happen? Now this inactivated repressor cannot bind to the operator. Okay, and now because it is not able to now bind to the operator, normal transcription and everything will take place. Now this ZYA, it will produce its beta galactosidase, permeases and trans-acetylase. So this is the first case when lactose, that is the inducer, is present. Okay, now what happened? The operator region is free because the repressor protein already got inactivated. Therefore, the operator protein is free. It allows RNA polymerase to bind to promoter. This in turn start transcription of Y, Z, Y and A gene. The necessary enzyme for lactose metabolism are produced after translation, and here we can say that the operon is switched on. So again I will explain what happened. In the presence of inducer, when there is a lactose present, what will happen? This will go and bind to the repressor. Now there will be some conformational changes and repressor will become inactive. Now because this repressor is inactive, it cannot go and bind to the operator. Now because it cannot go and bind to the operator, promoter is free to allow binding of RNA polymerase. Once the promoter will bind with the RNA polymerase, transcription will start. Transcription of the ZYA gene will happen. The mRNA, LAC mRNA will be formed. After that, there will be translation, and then there will be production of all the product beta galactosidase, permease, trans-acetylase. This is which is important for lactose metabolism, and this is what we call this operon is switched on. Now let's see the other part. Case 2, when inducer lactose is absent. Here you guys can answer. You guys will get the answer of NCERT exercise 10. In absence of lactose, repressor protein is free and in active stage. So now what will happen? The active repressor protein will bind to the operator region. So now what happened? Because there is no inducer, the repressor mRNA, which has formed this repressor protein, with the help of which this I gene, Now this repressor protein will go and bind to operator region. Now what will happen? The RNA polymerase will not be able to bind to promoter. It won't allow it to bind to promoter. Now there, when RNA polymerase is not going to bind to the promoter, then there will be no transcription. When there is no transcription, then there will be no translation and the no products will be formed. This binding to what? It blocks the RNA polymerase from accessing the promoter and transcribing what? The structural gene. And therefore, we said the operon is switched off. So earlier what statement we have gave, that inducer will decide this operator is switched on or off. That we have seen. If inducer is present, then it is switched on. If inducer is absent, it is switched off. And here the inducer is lactose. This regulation of lac operon by repressor is referred to as what? It referred to as negative regulation. Why? Because it has stopped transcription. And here I have explained you that what happened, this repressor protein go bind to operator, therefore not allowing RNA polymerase to bind to promoter, and then there is no transcription. So now let's come to the summary. If we talk about the translation, what is translation? Translation is the process of protein synthesis from mRNA. It occurs in ribosome, uses tRNA to deliver amino acid. 3-step initiation, elongation and termination is required. So what we have studied in today's translation thing, that how first we have studied that how the amino acylation take place, how the charged this tRNA will become charged. That tRNA when become charged means when the amino acid is attached to it, then what happened? Then the initiation step happen. In initiation step, what happened in of translation? Small ribosomal unit come and bind to the start codon at mRNA. Okay, now what will happen? The respective tRNA, the initiator tRNA, which is having this methionine amino acid attached to it, it will come, and that anticodon part, which is have the bases which is complementary to the codon part, it will come and bind to the start codon. Then what happen? Then the larger subunit of ribosome will come and bind to it, and this will form the initiation complex. So this is what initiation step. Then what happen? Just like that, the tRNA with their respective amino acid will start coming. It will start coming, and the polypeptide chain will start forming. But this is elongation step. But when the stop codon will come, we know there is no tRNA for the stop codon. So then will what happen? Then the transcription will terminalize. Okay. And how it is, and then the hydrolysis of bond between the polypeptide and tRNA occur. Therefore, polypeptide will become free, and eventually even the ribosomes will like detach itself, and during this process, when this stop codon will come, some release factor will come and bind to the stop codon. So this is the whole translation process. Then we talk about the gene expression regulation. Okay, it controls the protein production, timing and amount, involves promoter, operators and regulatory proteins. So we know what is the role of promoter, operators and regulatory proteins. If we talk about in this lac operon, what was the regulatory protein? That I gene, which was producing what? Which was producing the repressor protein. Whereas the operator is what? It is a DNA sequence that is present near promoter, and if the repressor protein will go and bind to the promoter, it will not allow the RNA polymerase to bind. If that repressed protein will go and bind to the operator now won't be able to give excess of promoter to RNA polymerase, and then there will be no transcription. Can occur at transcriptional or translation level. If we talk about lac operon, it is the classical model of gene regulation in bacteria. Control lactose metabolism. It contains structural genes, promoter and operator. It is regulated by glucose and galactose present. Functions through repressor protein. Okay. Now let's see some NCERT based question. First question is this, that which of the following is not a component of the lac operon? So we know that operator is the component of lac operon, promoter is the component of the lac operon, and structural gene is definitely the part of the lac operon. Which is not a component of a lac operon? That is enhancer So answer is enhancer. The lac operon consists of what? The regulatory gene, promoter, operator, structural gene. Enhancer are found in eukaryote, not in prokaryote operons. This is fundamental difference between prokaryote and eukaryotic gene regulation. Question number second. Which of the following statement accurately describe the role of tRNA in translation? Options are: it catalyzes the formation of peptide bonds, it carries amino acid to ribosome and recognizes the appropriate codon on mRNA, it provides energy for the activation of amino acid, or it forms the structural framework of ribosome. So we know that it carries amino acid to the ribosome and recognize the appropriate codon on mRNA. So the role of tRNA in translation is what? Amino acid are linked to their cognate tRNA. This is what we call charging of tRNA. During elongation, amino acid tRNA complex bind to the appropriate mRNA codon via complementary base pairing with the tRNA anticodon. Then peptide bond formation is catalyzed by ribosome. The energy for amino acid activation comes from what? ATP, and the structural framework of the ribosome is primarily formed by ribosomal RNA, not tRNA. So yeah, this is the answer for this question. Last question is that what happens when lactose is present in the cell? Repressor binds more strongly to the operator, repressor becomes inactive, RNA polymerase is degraded, or structural genes are deleted. So we know that what happens when lactose is present in the cell? Repressor became inactive. Events occur in presence of lactose. Lactose acts as inducer. It binds to the repressor protein. This causes a conformational change in the repressor, and what happened? It will not be able to bind to operator, and therefore transcription will take place. As I said, make repressor unable to bind operator, allowing gene expression, result in the production of enzyme needed for lactose metabolism. What are the keynotes for today's class? First, the protein synthesis required 20 plus different amino acids. 20 amino acids. One gene mutation can change entire protein function. Lac operon was first regulatory system discovered. Cells can make 2000 plus protein per second. Gene regulations occur at multiple levels. It could be transcriptional, post-transcriptional. Lac operon respond to glucose lactose availability. Translation uses over 50 types of tRNA. Here are some interesting questions that you guys are going to solve yourself. If a mutation occurs in the operator region of lac operon, making it impossible for the repressor protein to bind, what would happen to the bacterial cell's energy efficiency when grown in a glucose-only medium? Then the question is, why do protein synthesis inhibitor antibiotics like tetracycline tend to have fewer side effects on human cells compared to the DNA replication inhibitors? Third question, in a cell there are many more types of proteins than there are types of tRNA. How is it possible? Thank you. So here are the TPP question for today's class. First question is, which component of the ribosome act as a catalyst for peptide bond formation in bacteria? So we have studied it is 23S RNA. Save and next. What is the role of untranslated region UTRs in mRNA? We have studied that the role of UTR in mRNA is required for efficient translation. It doesn't got translated, but they are required for efficient translation. Then during translation initiation, the ribosome recognize which codons to start polypeptide synthesis. We know that it recognizes the start codon, that is AUG, which is also coding for methionine. Then what is the process of linking an amino acid to its cognate tRNA called? Means when we can say that it is a charged tRNA when the amino acylation happen. Then last question, which factor is responsible for terminating translation by binding to the stop codon? We know it is a release factor, which is, go and bind to the stop codon, and then, what happened? Hydrolysis of the polypeptide and tRNA happens. And translation stop. Even ribosomes get detached. Submit, yes. Here we can see all our options are correct. So it is all for today's class. Okay, thank you.